Hello everyone, and welcome to today's webinar titled Laboratory Tests for COVID-19. This webinar is part of the ongoing coronavirus virtual webinar series produced by LabRoots. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Jun Wu. Dr. Wu is an associate professor in pathology as well as vice director of Clinical Laboratory Medicine Center at the Shanghai General Hospital. He is also a youth member of Chinese Society of Laboratory Medicine, as well as a member of Shanghai Medical Association, Molecular Diagnostics Branch, and a member of Chinese Expert Committee of Experimental Medicine Pathology Branch. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Wu, you may now begin your presentation.
Hello everyone, I'm Jun Wu from Shanghai. 大家好，我是武军，我来自于上海交通大学附属第一人民院。It's such an honor to be invited by North American Chinese Clinical Chemists Association to share my thoughts and opinions with colleagues abroad on the laboratory tests for 2019 novel coronavirus. First of all, in order to better understand the rationale of the methods used for diagnosing the novel coronavirus in China, I want to briefly introduce the epidemiology and biological features of 2019 novel coronavirus. So these pictures displayed an outbreak timeline for COVID-19. On December the 31st, 2019, China alerted WHO about several cases of unusual pneumonia-like illness in Wuhan city. The pneumonia-like illness was named COVID-19 by WHO afterwards. Many of those infected were working at city's Huanan Seafood Wholesale Market, Huanan 海鲜市场. It was worth noting that Huanan Seafood Wholesale Market also sold live animals. 7 days later, the pathogen was identified as a novel virus belonging to the coronavirus family and was named 2019 novel coronavirus. Since January the 11th, the first death in Wuhan from the novel coronavirus outbreak was reported. More than 80,000 people have been infected, including 3,000 local medical staff, and 3,000 are dead nationwide. More than 40,000 medical staff from other provinces gathered in Hubei province, and most of them went to Wuhan city for treatment during the outbreak. So now coronavirus peak in China is over, as Wuhan has no newly infected cases, and new cases dropped to single digit in China mainland. But imported cases are continue to climb. But the overall epidemic situation remains at a very low level in China. But other countries such as Italy, here in Spain, report a sharp rise in COVID-19 cases, and it becomes a global pandemic recently. The impact of 2019 novel coronavirus has surpassed 2002 SARS epidemic in China and worldwide. The first genome sequence of 2019 novel coronavirus was performed by Dr. Zhang Yongzhen's lab at Fudan University its genome has about 29.9 kb. So the phylogenetic tree here is based on the nucleotide sequence of complete genomes of coronaviruses. 2019 novel coronavirus belongs to the subfamily of coronavirinae here. In the family of coronavirinae of the order Vidovirales. And its subfamily includes 4 genera known as alpha, beta, gamma, and delta coronaviruses. The whole genome sequence identity of the 2019 novel coronavirus shares a 96.2 similarity with one of the beta SARS related coronavirus strains, RATG13, that collected in Yunnan province, China, which did not list here, but more distantly related to its cousin, SARS coronavirus, about 79.6% similarity, and MERS coronavirus, about 50% similarity. So the 7 coronaviruses that can infect people are labeled in either blue or red. So there are 4 common human coronaviruses, 229E, NL63, which are alpha coronavirus, and OC43, which is beta coronavirus, and also HKU1, beta coronavirus, and 3 other human coronaviruses. MERS coronavirus belongs to the beta coronavirus that causes Middle East respiratory syndrome, or we call it MERS. SARS coronavirus, the beta coronavirus that causes severe acute respiratory syndrome, or call it SARS. SARS coronavirus 2, the novel coronavirus that causes coronavirus disease 2019, or we call it COVID-19. I'll list the receptor and the related disease of each human coronavirus here. Based on literature, the fatality rate for SARS coronavirus is about 10%, much higher than that of 2019 novel coronavirus, which is 3% at the beginning of the outbreak in Wuhan province. But recently, some countries seem to have much higher fatality rate. Anyway, initially the basic reproductive number R0 of 2019 novel coronavirus was slightly higher than SARS coronavirus. The person-to-person transmission route of 2019 novel coronavirus includes direct transmission such as cough, sneeze, droplet inhalation transmission, and contact transmission such as contact with oral, nasal, and eye mucous membranes, and aerosol transmission, which occurs when the virus droplets mixed in the air and form aerosol, and someone can be infected when breathed in. It also be transmitted through the saliva, and the fecal-oral dissemination may also be a potential person-to-person transmission route. Coronavirus are approximately 60 to 130 nanometer. It's a lipid bilayer that enveloped virion typically embeds 3 glycoproteins, the S protein, the membrane protein, and the envelope protein. The coronavirus was named for their chron-like spike projected on the surface. Inside the nuclear capsid, the N protein forms complex with genomic RNA, interact with M protein during transcription and assembly. The S, M, E, and N proteins listed here are all structured proteins. 2019 novel coronavirus was reported to share the same receptor, which is angiotensin converting enzyme 2, or called S2, with SARS coronavirus. By using the receptor binding domain of the surface spike glycoprotein S protein to engage SE2, but Daniel and his colleagues reported that S2 bind to the 2019 novel coronavirus S ectodomain with about 15 nanomole affinity, which is 10 to 20 fold higher than S2 binding to SARS coronavirus S protein due to 9 amino acid substitutions. The genome of 2019 novel coronavirus is a single strain positive sense RNA containing at least 6 OF arranged as OF1AB S E M N from 5' end to 3' end with 5' end cap structure and 3' end poly-A tail. The structure is similar to mRNA. The genomic RNA is used as template to direct translate polyprotein 1A and 1AB, which encodes non-structural proteins NSPS to form replication transcription complex RTC in a double membrane vesicles. Subsequently, a nested set of subgenomic RNAs, or we call it sgRNAs, are synthesized by RTC in a manner of discontinuous transcription. These minor strand sgRNAs serve as a template for the production of subgenomic RNA mRNAs. The genome and subgenomes of a typical coronavirus contain at least 6 OFs. The first OF 1AB, about 2/3 of the whole genome length, encode 16 non-structured proteins, except gamma coronavirus that lacks the first one. Other OFs on 1/3 of the genome near the 3 terminus encodes at least 4 main structural proteins, the spike proteins S, membrane proteins M, amyloid proteins E, and nuclear capsid N proteins. Besides these 4 main structural proteins, different coronavirus includes special structure and accessory proteins such as HE proteins, 3A and B proteins, and 4A and B proteins. All the structure and accessory proteins are translated from the sgRNAs of coronavirus. Patients with COVID-19 may start out with flu-like symptoms, including a fever, dry cough, sore throat, and shortness of breath, and so forth. There are few patients have little or no symptoms, or called asymptomatic infections. But the older patients and those with chronic medical conditions may become severely ill and even die as inflammation of the lungs start to spread and affect other vital organs like heart, kidney, liver, and so on, causing multi-organ failure. Hans Guang's lab identified through single-cell RNA-seq data analysis. The organs in red, such as lung, heart, esophagus, kidney, bladder, ileum, labeled in red are higher ratio of S2 expression, which is the receptor of the S protein of coronavirus, indicating the vulnerability of 2019 novel coronavirus infection. Since it is hard to identify suspected patients on their first visit to clinic because the initial symptoms of COVID-19 are similar to flu or the common cold, and also the high transmission rate of the disease. So the quick and reliable methods such as nucleic acid tests and immunologic tests are widely used in clinical laboratory for screening out those people infected. Nucleic acid tests, especially the fluorescent PCR, is considered as a gold standard for diagnosis of COVID-19. But false negative results are not uncommon. Professor Wang Chen, he is an academician of Chinese Academy of Engineering. In an interview with CCTV on the 5th February, estimated that the detection rate of nucleic acid test for positive patients was about 30 to 50%. It means that half of the real COVID-19 cases were missed, and infected patients could go unnoticed, which made the situation in Wuhan at that time even worse. So the diagnosis and the treatment program of 2019 new coronavirus pneumonia, the trial fifth version, now is 7th edition. Now the 7th edition has been released. This is a 5th version by National Health and Medical Commission of China in 2020. Allowed cases only in Hubei province to be diagnosed clinically, such as CT images showing characteristics of vital pneumonia. So on February 13th, 2020, more than 13,000 new clinically diagnosed cases were abruptly added to its total count in Hubei, leading to a sharp uptick in total confirmed cases. Chinese Society of Laboratory Medicine published the expert consensus on nucleic acid detection of 2019 novel coronavirus on Chinese Journal of Laboratory Medicine. The early February to direct the doctors and technicians in the epidemic areas how to protect themselves during taking samples from patients and transferring samples to the lab, and also how to handle different types of specimens with extra care, and how to improve the detection rate. And I think American CDC also has its own guidelines. So in the consensus, the principle for the safety are you need to fully covered to protect yourself from the virus. Level C here meaning additional precautions, including personal protective equipment such as protective clothing, surgical masks, the face shield, and other physical barriers like a splash shield. So tests including nucleic acid tests and immunological tests should be performed at least in BSL2 laboratory. The nasopharyngeal swab, oral swab, sputum, bath, blood, and stool specimens are suitable for PCR test. So the specimens should be sent to the lab. The sooner the better. But for long-distance delivery, you need to obey those packing and transportation rules in China. I think it is also required in the US. If it takes too long during shipping, for example, longer than 72 hours, you need to put your specimens at -70 or even lower temperature. For the blood samples, Plasma need to be separated from the whole blood before transportation and storage. The specimens should be fully sealed in containers, and the labeling should be clear when delivered to the lab. For specimen processing step, the virus inactivation can degrade the nucleic acid of the 2019 novel coronavirus, resulting in the decrease of the CT value and produce false negative results, especially for the low concentration specimens. Also, it is recommended to apply automatic nucleic acid extraction equipment using magnetic beads to get maximum nucleic acid recovery rate. To interpret the fluorescent PCR data, we need to refer to the manufacturer's instructions. Here's just one example for the 2019 novel coronavirus detection kits. Actually, most detection kits use the same strategy that target two sites on 2019 novel coronavirus genome. One is on OFF 1AB and the other is on N genes. Only the CT value of both sites are lower than 37. We can conclude that 2019 novel coronavirus is detected. Many steps that would affect the result of a nucleic acid test, but here I want to stress the importance of the following steps that often get overlooked. First, the cause of disease should not be ignored because it would become a great disparity for viral load of different specimen sources at different stages. Yang found that the viral load in throat and sputum samples collected from the 80 patients at early stage after disease onset was at a higher level, and the viral load in sputum is higher than the throat swabs. So analyzed the viral load in nasal and throat swabs obtained from 17 symptomatic patients in relation to days of onset of any symptoms. Higher viral loads were detected after symptom onset, which is consistent with Yang's finding. The higher viral loads were detected in nose, then in the throat. Conversely, the viral load in patients with SARS normally peaked at around 10 days after onset, suggesting the more transmission potentiality of patients with COVID-19 than ones with SARS at the early stage of onset. Yang also collected different types of specimens from 2019 novel coronavirus confirmed cases, and those cases divided into 3 groups based on the collection time, including the 0 to 7 days after onset, and 8 to 14 days after onset, and the longer time, 15 days after onset groups. They found at the early stage after onset, the lower respiratory tract specimen, the sputum, has a higher detection rate than nasal swabs, and the throat swabs is the worst specimen sources. For the critical patients, the lower respiratory tract specimens, the BAF, can get a higher positive detection rate. Here's 100%. The BAF is better than the sputum, and the sputum is better than the nasal swab, and nasal swab is better than throat swabs. For the recovery stage, they find that nasal swabs much better than the throat swabs. So although BAF specimens is considered very accurate, it should be obtained from severe illness patients receiving invasive mechanical ventilation. The deep coughing sputum is another recommendable specimen. It showed lower detection ability only during the recovery stage of mild illness, but still better than the throat swabs. But only 28% of patients have sputum production, but the other had only dry coughing. Secondly, the performance evaluation of nucleic acid amplification tests should not be neglected, especially for the lower viral abundance samples. The South Korean research group compared the primer and the probe sets for 2019 novel coronavirus that developed by CDCs in China, Germany, Hong Kong, Japan, Thailand, and also USA. The sequence of primer probe sets for each country are different, and their locations at a viral RNA genome are showed here. The China CDC suggests the OF1AB and N. The Germany uses RDRP SAS-SR. The RDRP stands for the RNA-dependent RNA polymerase, and also the E gene. The Hong Kong use HKU N, and where it is? Oh, here, the HKU of 1B, here. And US CDC uses the N1, N2, and N3. Thailand use here WH-NIC-N, and Japan use this primer set. They find that in the case of targeting RDRP region, this region, the China OF1AB set might be the most sensitive than the others. So the US N2, N3, and Japan N2D, this sets may be recommended for sensitive qRT-PCR assay for N region. So in the market, the China government also approved 7 detection kits with a fluorescent PCR method. And more than 100 related nucleic acid diagnostic products are applying for the certification from CFDA, meaning they are currently for research use only. Guo and colleagues compared the detection performance of different 2019 novel coronavirus nucleic acid detection kit. They find that there are differences in detection ability of six 2019 novel coronavirus nucleic acid detection reagent for weakly positive samples, and their accuracy, sensitivity, and reproducibility of some reagents are not good. Why is that? Firstly, the amplification system are different. Most of the products M2 targets on its RNA genome, which are OF1AB and E or N genes. But the triple target genes detection kit, or even 4 target gene detection kits also have their market share, probably because they present better specificity and sensitivity. Secondly, among 10 mainstream double target gene detection kit, they have different sensitivity according to their instruction sheets. The higher sensitive one, such as the ones that could detect as low as 100 copies per mil viral RNA, could have an advantage on weekly positive sample detection. Immunologic tests such as total antibody, IgM, IgG test are valuable supplement to nucleic acid test. On early and progressive stages, which are arbitrarily set 1 to 7 days after onset, the combination of immunologic tests with a nucleic acid test greatly improved the overall detection rate of 2019 novel coronavirus, particularly for those patients with negative PCR result. On late stage or recovery stage, antibody test surpassed nucleic acid test. Genetic analysis identified mutations in 149 sites across 103 2019 novel coronavirus strain genomes. The sequence similarity is about 99.5%, suggesting that the 2019 novel coronavirus genome is still stable. This virus evolved into two major types, designated L and S, that are well defined by two different SNPs that allowed that showed nearly completely linkage across the virus string sequenced to date. This analysis further supports the idea that the two linked SNPs at the site here and here adequately define the L and S type of 2019 novel coronavirus. The mutation site of S and L subtypes are not commonly used for 2019 novel coronavirus nucleic acid detection and antigen design. Normally, we use N protein and S protein for the antigen design. So the impact on nucleic acid test, immunological detection, and even the vaccine of 2019 novel coronavirus is minimal. Lastly, I want to discuss about the circumstances that nucleic acid test showed virus infection again for some discharged patients, which has not only happened in China, but also found in other countries such as Italy, Spain, and Japan, etc. Patients in China are discharged after 2 negative nucleic acid tests, taken at least 24 hours apart, and indications of clinical recovery, including the resolution of symptoms according to the new coronavirus pneumonia diagnosis and treatment program, trial version 7, which is the latest version by National Health and Medical Commission of China. But the Lan reported in JAMA, they wrote the RT-PCR tests were repeated 5 to 13 days after hospital discharge or discontinuation of quarantine of 4 patients, and all were positive. All patients had 3 repeated RT-PCR tests performed over the next 4 to 7 days, and all were positive. The worrying cases of recovered coronavirus patients in China testing positive again for the infection have raised questions among doctors about the criteria being used for discharging people from hospital. So some hospital I know in China have adopted 3 negative results as a discharge standard. But even so, currently there are still many cases testing positive after patients being discharged from hospital. There would be, I think, 2 possibilities for these circumstances. So in China, the nucleic acid test of throat swab was used for evaluation the condition of recovering patients. However, for those patients in recovery stage, positive rate for viral nucleic acids in throat swabs is much lower than nasal swab, which I showed in previous slide. Hu and colleagues reported in 2 of 3 cases of COVID-19 with respiratory tract infection and no gastrointestinal symptoms. The anal swab specimens retained positive 6 and at least 14 days after virus turned negative in the respiratory tract. And in the rest case, the virus nucleic acid turned negative in anal and nasopharyngeal swabs on the same day, indicating that the stool is a better specimen than nasopharyngeal swab for evaluating the conditions of recovering patients for nucleic acid tests. The similar findings also happened on patients with SARS coronavirus. It is less likely that discharged patients feel ill with the virus again. But if convalescent patients, especially the old patients and the ones with chronic medical conditions who did not build up enough antibodies to develop immunity to the virus, they will be in high risk to get infected again, because the dormant virus by the antiviral the treatment could create new symptoms after this isolation. There are 2 ways to settle the issue, I think. The first one is monitor 2019 novel coronavirus antigen and the specific IgG levels. Patients with detectable antigen or a low level of IgG should be retained in isolation. The second, the quarantine for 14 more days after discharge. Okay, this is the last slide. I want to summarize all those information. For the early stage of the disease or at the time of hospital admission, nasal swab, throat swab, and sputum sample are priorly recommended for nucleic acid test. During the critical stage, if nucleic acid test for nasal swab and throat swab have negative results, the sputum and the BALF sample would be the best choice. For monitoring the patient's recovery, stool or anal swab are preferred. In order to improve the detection rate, antigens and antibodies should be tested alongside the nucleic acid test. For more information about this table, check out this link. It was a review I wrote for Chinese Journal of Laboratory Medicine last month. So that's it. Thank you.
Thank you, Dr. Jun Wu, for your informative presentation. As a reminder, our speaker will follow up with any questions you have submitted via email following this webinar. We would also like to thank LabRoots for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we do not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information he provided at the time of registration. This webcast can be viewed on demand through the end of this year, 2020. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.